Hello, I'm Tim McClanahan. I'm representing a large group of people working on a project to identify hotspots in the Western Indian Ocean. And this slideshow was created to keep you informed as to the progress of this project. If you've seen a previous recording, it's probably changed since then. As we learn and do more things, I'm adding slides in order that by the end of this investigation, we'll have a full presentation of all the research and conclusions and recommendations to date. As a reminder, this part of the biodiversity analysis that I'll describe is part of a larger workflow to evaluate the institutional analysis and the policies, laws, and regulations in order that we can combine those to develop these priority focal areas. Once we do this, we'll follow with a situation analysis, which gives us a view of the threats to this biodiversity and possible actions, and then we use that to make recommendations. One thing to remember about estimating biodiversity is that they are very methods dependent, and these methods may have some limitations to estimating areas with high biodiversity. One of the main methods shown on the left is using presence-absence data, in which sites with these species present are connected often by polygons, and then layers of polygons are added on each other to estimate the total amount of species or diversity in these particular sites. One of the problems with this approach is that there are large areas in between presence-absence observations where these species may not occur. The result is that you get very coarse scale estimates of biodiversity, and these overlapping distributions sometimes tell you more about the between-site diversity than the local diversity. That's a problem for identifying particular local hotspots, which are areas where there is dense local and resident diversity. Another approach shown on the right is one where a certain number of study sites are evaluated in the field, and then what are called rarefaction techniques are used to estimate biodiversity on a larger scale. This method is based on local sites, but in many ways it's extrapolating diversity beyond the site with these rarefaction methods such that you can estimate diversity on a larger scale. Again, it has a scale problem where in many cases you're extrapolating beyond the local site to estimate some sub-regional or regional biodiversity level. Both of these approaches often lack local heterogeneity, and that's a problem for the scale at which we'll be searching for biodiversity. In many cases, we're interested in biodiversity scale at the tens of kilometer area rather than the hundreds or 1,000 of kilometer area. So in many cases, the scale of management, such as at marine protected areas, is often at the scale of 10 to 50 or 100 kilometers of coastline. And therefore we need to develop new techniques that will allow us to identify biodiversity at that particular scale. Here are some of the people that have collaborated so far sharing their data. You can see the names on the left, their institutions, the countries where they've contributed data, and then the types of data they've contributed. This has led to a fairly large data set that represents considerable coverage or distribution in this region. Here's some of the summary information from that biodiversity data sharing. We have about 1,000 observations of coral, mostly at the general level. We have about 1,200 observations of fish at the species level. We will be using the families named here to act as a proxy for total species richness. We've combined at least 61 environmental human variables that I'm aware of, and there may be a few more. And we're using machine learning statistical methods to develop models to fit to the biodiversity data, that environmental data, and that is being then used to fill in biodiversity estimates based on the environmental data and the approximately 7,900 cells without data. Here you can see the distribution of the empirical data. You can see it has good coverage. The data is collected between 1991 and 2002. The data is pooled into these 2.5 by 2.5 reef cells to correlate with the environmental data at that spatial scale. We've looking at a number of variables. I'll show one interesting variable at the end here of the effects of time on the biodiversity of corals and fish. This is just a reminder of what the main ecoregions are in this location. The East African ecoregion and Madagascar are the two largest ecoregions. And we've also, all of the cells are divided into 4 fisheries management categories based on experience of ourselves and other observers. And all of the 60-some environmental variables are mapped. I picked a selected group here to show you. But each of these are mapped, and we can see where those variables vary across the region. This is a correlation matrix of the environmental variables and their distributions. What we do first is look for variables that are strongly correlated with one another in order to make decisions about which one should be included in the final model or in the final models. And we use this as a way to avoid putting redundant variables in the model, and also to look at model options. So in this case, I'll be showing 2 models, one where we use the top most highly correlated variables and then eliminate the lesser correlated variables, and then the second model where those lesser correlated variables are included in the model. This gives us an opportunity to look at how the model might be changed based on the variables that you selected. But what we do is we finally take the average or combination of the 2 models to develop the ultimate final model for the predictors of biodiversity. First, I'm showing you the results for the corals. These are the 2 models after developing alternative models. These are the results after tuning the model parameters to reduce deviance. And you can see these are the main drivers of fish biodiversity by these 2 models. You can see first that fish biomass is the single largest and a great contributor to biodiversity, but other variables such as travel time and SST and other variables that you can see listed here also contribute to the model for total biodiversity. In this case, I believe there are about 26 variables that contribute to the final prediction of biodiversity of fish. Here you can see the important variables for coral. The observer effect is fairly strong in corals, but there's a number of other important local variables such as depth, but also what might be considered environmental stress variables. Here you can see the responses to each of the variables. I'm showing the top 9 variables here. You can see the biomass is a saturation response, and saturating somewhere close to 1,000 kilograms per hectare. But most of the biodiversity is explained by the first 600 kilograms per hectare. You can see the observer effects are not particularly large in the fish. And you see a number of other response curves here. In Model 2, where we retain some of the connectivity metrics, they turn out to be fairly important for biodiversity, or at least they contribute to the overall model. Here are the response curves for corals. You can see that observer effect is there, but there are another number of important ecological responses. Diversity is maximized as about 10 meters. Degree heating weeks, cumulative degree heating weeks is sort of a hump-shaped relationship, maximized at a modest 20 cumulative degree heating weeks, and a number of other variables that indicate negative effects of skewness, very high kurtosis, but also effects of dissolved oxygen being higher, diversity in low dissolved oxygen, low salinity, and low current velocity as important variables. You can see here then from the models we're able to make predictions in all cells in the Western Indian Ocean. We have to keep certain things constant, for instance depth and biomass, and we pick observers with the highest replicates per country, and that allows us then to make predictions for, in this case, fish species for those 7,039 cells. We combine, as I said before, the two models to create this final maps. On the left you see coral diversity, distribution showing high diversity along the African coastline and in northwest Madagascar and some of the offshore islands. Lowest diversity down in the middle southern parts of Madagascar. On the right you see the distribution, the predicted distribution of fish diversity. Again, high diversity along the African coastline, southern Kenya down to northern Mozambique, with also high diversity in parts of some of the oceanic islands in the northern parts of northwest parts of Madagascar. And then the final map is a combination of the fish and coral diversity patterns. You can test the models against the data. If you do a model based on 70% of the data and test it against the remaining 30%, you'll see you get pretty high R-squared, or predictions for the fish, as high as 0.64. If you look at the full model and not use independent data, you get very high fits of 0.83. When you look at coral diversity, you get somewhat lower fits for the 70-30 comparison, around 0.5. But then if you look at the full model, you get fairly high fits at around 0.854. So these are generally very good predictive models and give us some indication that the predictions are probably pretty accurate. They will be less accurate as you predict what's happening in the future, but at least for the current state, they are quite accurate. Here I'm showing the relationship between the coral and fish biodiversity. On the left you're seeing the predicted from the model, and on the right you're seeing the actual empirical data. So the points here is that they are positively correlated, but the second point is that there's fairly high variability, that they're not a tight correlation, such that when you combine these two sources, you're probably covering different aspects of the forces behind the biodiversity, and therefore probably getting a good indication of what biodiversity is like using these two proxies. The combination should be fairly effective at identifying locations with high biodiversity, recognizing that different assemblages will have different responses to environmental variables. One of the things we're doing is not just looking at the biodiversity at the provincial level, but also looking at the country level. So here you're scaling biodiversity by the diversity within that country. So you can see within each country where the high and the low spots are located. This particular map brings the high diversity sites to the fore, where cells are close to each other, but allows you to give you some indication of biodiversity independent of the provincial patterns. You can see here that there are some differences, and when you compare a country, you get different responses than you might get from a provincial level analysis. There are some interesting things you can do with the data. The individual responses of your coral diversity to various metrics is interesting on its own, and something I'll propose as one of the papers. But you can also look at things like what is the effect of the sampling date on the predicted species richness. And here you're seeing it for fish on the left and corals on the right. Sampling date was not a strong predictor, but you can look at the model's predictions nonetheless, and you can see here that there is indication that fish diversity is declining but fluctuating, possibly associated with the 1998 El Nino coral mortality event, and possibly with some similar coral mortality in 2016 or other types of stresses. The coral plot seems to suggest there's sort of a declining, stepwise decline in coral diversity over time in this region. Now we get to one of the main points of this exercise, is to determine where the biodiversity hotspots are located. And what you can see here is it depends on the number of criteria that you use to designate hotspots, and also on the spatial scale or the spatial characteristics of the hotspots. So you have 3 measures of biodiversity, coral, fish, and they're combined, and you have 4 scales. One is the province, one is the ecoregion, and one is the country. And then we have a 4th scale, which is what we call local clustering. That is the density of spots within this 41-kilometer step length. And this tells you where there are areas of dense high biodiversity within these 6 square kilometers. So each of these has a slightly different prioritization, whether you're looking at the whole province, the ecoregions, and the countries, and whether they're clustered or not. This would be an example for the whole province. These would be the top sites, many of them fitting all 12 criteria, some of them a few less. But what you can see then is there's a band of provincial hotspot priorities that runs from northwestern Madagascar across to Mayotte, Comoros, the Lazarus Banks in Mozambique, and then quite a few sites along the coast from southern Kenya down to mid-level of Mozambique. When you look at the ecoregional hotspot distributions, you can see some changes. There's still many of the hotspots you saw on the East African coastal current along the African coastline. But when you now look at the specific ecoregions, you can see the Seychelles has its own hotspot in the Fakwatir Islands. There's a remote ecoregion called the Karagos-Tromelin Islands, northern part of Mauritius, and there's islands there called the Agalega that's identified as a hotspot. So you can see these changes. It's particularly interesting in Mozambique, where they have 3 ecoregions, and that causes more of the hotspots to be located further south, where these 2 additional ecoregions are located. Also in Kenya, Kenya has 2 ecoregions, and the East African coastal current shows the hotspots in the southern area on the border with Tanzania, but the East African coastal current is located in the north, and that creates hotspots specific to that ecoregion in the Lamu area. So changing scales has this effect, and I'll show another example of that when we scale at the nation. When we scale at the nation, you can see that some of these remote islands, such as Mauritius and Reunion, have their own specific hotspots. You can see Mauritius on the upper right there has at least 3 hotspots at that particular region, and then unsurprisingly Reunion has a hotspot on its particular coral coast on the leeward side of Reunion. Some of the other hotspots are still there for the Seychelles, for example, but there's some other changes. For example, quite a few additional hotspots in the northern part of Madagascar that were not seen at the provincial level or even the ecoregional level, but at the national level, there's considerably more hotspots. In the case of Kenya as well, you can see that the hotspot area moves further to the north to include areas up into the Diani area as hotspots because of the restriction of hotspots to just the area in Kenya. So at the national level, you get a slightly different view of these particular hotspot areas, and this is important for national level planning. Finally, if you look at our method of clustering neighborhood sites, looking for neighborhoods of densely high biodiversity areas, you can see here that this results in elimination of many sites in the offshore of the African coastline and gives results more similar to the provincial level. And this is due to the high density of high diversity sites along the African coastline, again from southern Kenya down to northern Mozambique. These areas are areas where the reefs are within 6 square kilometers with high diversity are located close to each other. So these are focal areas that will include more than one reef, more than one high diversity reef that have a cluster of high diversity reefs. This work is ongoing, and I'll continue to present updates on this work as we go along. Currently we're working to identify the top 21, 9 hotspots, and particularly identify 9 hotspots for further evaluation. We're getting input from the USAID missions to determine what their priorities are in order that when we go to the focal area analysis, the threat analysis, or the situation analysis, that we'll have their confirmation about the important 9 hotspots for this work. Within the modeling framework, we can further do experiments with the model. We can examine certain counterfactuals. For instance, how might biodiversity look in the absence of important controls such as fisheries management systems, the effects of people. There are other types of scenarios that we may be able to investigate with these types of models. Additionally, this work has focused on numbers of species as a proxy for total numbers of species, but there may be some interest in evaluating other aspects of biodiversity, such as the community structure, the community composition of these communities throughout this region. We are now slowly developing the scientific publications that we expect to come out of this group. We have 3 outlined at the moment, and we'll shortly be in the process of getting feedback from our collaborators and working on finalization of these 3 proposed publications. Some more details are given in the following slide for those people that are involved in publications, but this concludes this particular informational part of the slideshow. Thank you very much for watching. Paper 1 is aligned most closely with the goals of this paper, which is the identification of biodiversity hotspots. And in this case, it's to do it irrespective of the specific taxa, but to combine the 2 taxa and the various models, develop our best maps of biodiversity with the coral and fish and their combinations as indicators of total biodiversity. So the point here is that to create sort of the overview paper and to move with this fairly quickly, because it aligns very much with our reports for this particular proposal. And the idea here would be to look at various distributions, maps, possibly at different ecoregions, but mostly to present the relationship with the environmental variables and then the predictions for all cells, possibly to look at provincial, ecoregional, and national levels, and to come up with hotspots for each of these specific levels. This would involve data contributors from both corals and fish, and work as a larger team on this, whereas what I'm proposing for the other two papers is divide the data into just the coral and just the fish in order to bring out more information about those two groups. The idea of this paper would be that they should be fairly short, a limited number of elements, tables, and figures, and get in and submit it to these journals such as Diversity and Distributions that have fairly limited word limits and element limits, and that required us to sort of focus on a few key things. The more details of these papers are given in a Dropbox in a file that you can go through and comment on at the moment. But here's our highest priority paper for the moment. Paper 2 would focus on the coral communities in the Western Indian Ocean, and the idea here would be to model not just numbers of coral species, but also coral cover and some multivariate axis of community structure, to look at how each of these four variables responds to the environmental variables with the BRT method, and to look at the correspondence between these variables among each other in order that we get a broad view of how communities, species, and cover are changing across the region and how they're interrelated. Here we can pick some counterfactuals to determine what might be the future change due to population growth, or perhaps what distribution might look like in the absence of people. There's a number of scenarios that we can consider here. These kind of papers will need to be short format and focused, and that'll allow us to submit papers to some of the top biogeography journals. Paper 3 would be very similar to paper 2, but here we would focus on fish ecology. The idea is to have maybe 4 response variables again, numbers of species, fish biomass, and multivariate community structure, and to look at how each of these variables responds to the environmental variables and the BRT methods, and to look at the correspondence between these variables as you look at these gradients of environmental influence. We can choose some scenarios as well if space permits, but the idea is to have a very fish-centered paper, which will give us a good idea of how these variables, 4 variables, are distributed in the Western Indian Ocean, and what are the environmental variables associated with them. I just wanted to finish by reminding you that the other part of the study, which is on governance, is a questionnaire-based study. There's a institutional analysis questionnaire, which is 8 questions for institutions that are involved in biodiversity management, to tell us a little bit about what their information is and plans for climate change are. And then ultimately there'll be a situation analysis for the focal areas, which will depend on what the focal areas are chosen, which we should come to very shortly here. I wanted to remind you that if you work with people that are involved in biodiversity management, and they should be answering these questionnaires, please contact us. And if you can, help promote the questionnaires being completed. Getting people to, as you probably know, to complete questionnaires is a challenge, and it helps if they know what the purpose is and if other people in the region care about the responses. So you may be able to help us here in identifying locations. These are some examples of participating organizations that we have to date, but we hope to fill this out and get responses from these groups in the next few months. So thank you very much if you're able to do that. These are the main people working on this project within our office. That's me, and Maxwell is the statistician doing much of the modeling. Nyawita is doing much of the governance institutional work, and working closely with Carolina Bungay. If you have ideas or contacts or other types of information that might be useful to us, these are the people you should be contacting.